Welcome students. This is our class for Nutraceuticals lecture 4. Here we are going to discuss about an interesting topic, nutraceuticals in cancer treatment and in cancer we can say. So we know at present cancer is the biggest threat for the mankind. So we can see what is its application of nutraceuticals in cancer treatment. We have discussed in the first class itself about nutraceuticals, which are more useful in prevention and as well as for treatment of any ailments, especially chronic diseases. We have already discussed about diabetic mellitus and cardiovascular diseases. This picture just to show you that the anti-cancer plants and its phytochemicals having the application in preclinical cytotoxic assessment of cancerous growth of tissues and cells, where you can see the cancerous conditions of a human being or a patient, treatment which is given by clinical therapy, by chemotherapy and radiotherapy. All the therapies can be accompanied with phytochemicals from various anti-cancer plants. Apart from that, today our discussion is nutraceuticals in cancer. So the need of nutraceutical is due to why? What's the need of nutraceuticals and cancer treatment as well as prevention? Why? The question give the answer that the leading cause of death of adult in cancer. Treatment of cancer involves expensive and traumatic use of drugs, surgery and irradiation. This all makes the need of nutraceuticals, and there has been vast growth of nutraceutical industry to yield large number of phytochemicals containing nutraceuticals with diverse composition and healthy or health claim due to increased application of phytochemicals in cancer care. So you should know that the increased application of phytochemicals in cancer care has been vastly increased. The arrow denotes that it has been increased. That's why the need of nutraceuticals becoming a biggest demand in the world. So this is just to have an information about how a cancer, not in detail, just an outline for a students to know how a cancer is developed, cancer formation, just an idea. So normal cell, it gets DNA damage, how the DNA damaging agents like chemicals, radiation and viruses, they can damage the DNA and fail DNA repair. And this failed DNA repair can be due to gene affecting DNA repair and mutation in genome. Mutation in genome is by gene affected in cell or gene affection in cell growth and reduce apoptosis, and this promote oncogene. So you can see suppression or suppressed gene causes to unregulated cell proliferation, excess clone formation, unwanted cell division, tumor formation, then causes the death. This is a simple idea about cancer formation. And even after mutation in gene, regulate apoptosis and decrease apoptosis, excess clone formation, tumor formation and death. So this is a just idea. We should know how cancer is formed in our system of body and where our nutraceuticals can support in the form of care in cancer. So, nutraceuticals have the ability to control and regulate the DNA damaging factor in cancer cells and DNA transcription in tumors. The herbal-based dietary supplements, we can say that is nutraceuticals known to antagonize the therapy for cancer. The research says that phytochemicals are proven to be the potential to interfere even with the latest age. That means we say primary, secondary, tertiary conditions of the cancer. So latest stage, the final stage cancer, and altering the metastatic spread of cancer. So it has lots of evidence to support the cancer treatment. Numerous nutraceuticals have shown to regulate specific tumor suppressor genes and cancer driving genes. So here in this slide we are going to see a point which students can write in the examination: promises of nutraceutical in cancer research. What all the promises? We can write by points. First one is, it gives pleiotropic activity. The research of the nutraceuticals in cancer and research, it shows it has application in pleiotropic activity. We should know what is this pleiotropic activity. Inhibition in the various enzymes, we'll see in slight details about pleiotropic activity, how it inhibits the enzyme proteins which are responsible for the cancerous development, and target classical signaling pathways target classical signaling pathways. And number 3, these nutraceuticals can sensitize tumor cells to chemotherapy, radiotherapies which were given for cancer treatment. And generally the nutraceuticals can well tolerated for the cancerous developments and to control the cancer cells formation. And novel synthetic analog for improving efficacy. Novel synthetic analog, which we know that how the herbs are applying in the novel synthetic analogs in delivery systems, which we know that many nano particles, dispersing particles, and many type of liposomes which are used for novel drug delivery systems can also be developed using and supplements of nutraceutical formulations. First, we should have a discussion about the pleiotropic activity. Just now we have told the first challenge, the first application or first giving evidence by the nutraceuticals, which is that by natural sources which can give impact of anti-cancer by phytochemicals after inhibiting expression of various genes, proteins, enzymes and signal cascades in order to block cancer initiation and progression cells in our body. So this is a picture just to show about how mushroom constituents, lycopene from tomato and curcumin from various phytochemical constituents influencing the cell inside. You can see P16, P21, E1K1, these all numbers and specified enzymes, that is cascades enzyme 384, how they can control over and inhibit this to control the formation of various tumors, and various cell divisions can be controlled. And this is called this pleiotropic activity of the phytochemicals in the cell divisions. So students can write very clearly, understanding by means of pleiotropic activity given by the nutraceuticals or the phytochemical constituents from the herbal products or herbs can have an impact of anti-cancer activity. And the next one is, you can see, targeting the pathways. You can say how it is can be target signaling pathways. Targeting signal pathways by natural products by induction of autophagy, by inhibition of angiogenesis, by induction of apoptosis, and acting on the cell cycle arrest and increase the CDK inhibitors and decrease the cyclins and inhibit the EMT, and you can see that inhibition of pro-inflammatory cytokinins. So all of them, they makes the inhibition of pro-survival signal pathways and suppression of tumorigenesis. So tumor formation have been inhibited on the whole. The signaling pathway, the targeting pathway have been inhibited by the natural products, that means the natural source through the herbal products, through nutraceuticals in cancer. This is the second point. The third one is sensitizing the tumor cells by the therapy which is given by radiotherapy or chemotherapy to the cancer treatment have been sensitized by nutraceuticals. So you can see the natural compounds, nutraceuticals, can elevate the toxicity of chemotherapeutic drugs which is given. It can reduce ionizing radiation danger to normal healthy cells, which can reverse or prevent, both reverse and prevent the tumor cell formation and radio and chemo resistance in tumor cells, like DNA damage, inhibition of cancer cell survival pathways, and reducing the cancer development by cell death, promote or potentiate the anti-tumor activity. So this is also by natural compound nutraceuticals. And the next one is, the fourth one we can see, that is generally well tolerated. The cancerous treatment or cancerous cell by initiation, propagation, promotion and progression. This is the main things which is happening in a cancer, like normal cell, initiation cell, uncontrolled cell growth, and then can tumor metastasis will take place. But our nutraceuticals like phenols, just an example we are given of buckwheat, as act as an antioxidants, can be inhibited the formation of initiation cell, and hydrated of papain or chia papain, which can have an inhibition in inflammatory activity, which initiation cell can be inhibited of proliferation, can be inhibited in the promotion. That means initiation, you can see inhibition of inhibitory activity, stimulation of DNA repair can be inhibited by phenols, and in induction of cell cycle arrest and induction of apoptosis, inhibition of cell propagation, proliferation can be done by hydrated of hydrolytes or hydrolysates of say papain. And then you can see when you are having uncontrolled growth, that place which we can say progression area, in which you can see down regulation of MMPs and angiogenesis, inhibition of prolifery and metastasis can be done by peptides of buckwheat anti-proliferative. That is progression stage. So initiation stage, promotion stage, progression stage in cancer can be tolerated by means of nutraceuticals. This is what we should have an understanding in cancer application. And you can see the parent structure unable to design, as you can see the novel design of analogs of release of drug improving the efficacy can be done by cancer in novel drug delivery system of management, like we can use hydrogels, the synthetic can be done with along with the natural sources, like preparation of drug delivery system like hydrogels, microparticles, measles, exosomes, radiotherapies, and you can see microbubbles and liposomes and dendrimers. Many delivery systems can also support in the form of novel drug delivery system in management can be done with the help of nutraceuticals. So next one is challenges. There are some challenges which are coming across in preparing nutraceutical in cancer treatment. What are they? First one is bioavailability. Bioavailability plays a major role for any action or any absorption, distribution. After this, all things happen, the kinetics, the dynamics depends upon the bioavailability. So pharmacokinetics, pharmacodynamics, which you come across in your subject, the dynamics, the activity of the drug depends upon the bioavailability of the drug molecules in the blood. So that's bioavailability metabolized act effectively high systemic concentration, difficult to achieve. And then insufficient preclinical data is also giving some difficulty challenges in research of nutraceuticals in cancer treatment. And insufficient, that means insufficient preclinical data, advanced combination multi-agent therapy in clinical setting. And number 3, nutraceutical may not be patent. That's why low priority is given by pharmaceutical industry as they may not be patent. And things which you are going to, very important thing, sometimes they may ask you, give some nutraceuticals more active as in cancer treatment and cancer prevention. That means we should write about these slides, these points. I have taken very little nutraceuticals. There are many. Number 1 I have taken, that is chestnut. Number 2 is berries. Number 4 soya. Number 3 soya. Number 4 garlic. And number 5 green tea. Number 6 grape seeds. Number 7 ginseng. And number 8 Rhodiola roseae. And number 9 tomato and red pepper. Number 10 dietary fibers and cruciferous vegetables. So these all 10, there are many, but still we have taken only 10 examples of nutraceuticals, functional foods as a nutraceuticals in bioactive compounds in cancer. So we can go for the first one, functional food example. The first one, chestnut. Chestnut extract possesses antioxidant activity, and it protect you against gastric cancer, and mild protective effective against prostate cancer and breast cancer. The chestnuts are a group of 8 or 9 species of deciduous trees like this, and shrubs in the genus of Castanea. They are shrubs in Castanea. In the breach of family Fagaceae. So you can see the fruit when it is open. You can see the nuts. This is fried one. It will be very tasty and delicious. And it has more vitamin C actually. It's not much available in India, but it is in Europe and as well as in Asian countries. In Arab countries they are available. So it is very interesting nut which has more nutrient and anti-cancer activity, specifically prostate cancer and breast cancer. They are having action. They have mild action in gastric cancer also. Berries. We know many blueberries, blackberries, strawberries are the good source of antioxidants and phenolic compounds. This indicates that berries may be effective in the prevention of development of cancer cells. This is blueberry, blackberry, and strawberries. You may be knowing these are strawberries A repository of bioactive phenolic compound has been investigated for the contribution of their antioxidant activity to prevent the chronic diseases. Blueberries have been found to be chemo preventive to breast cancer in vitro and in vivo studies. Soya, in research the soya, the risk of cancer must attention and interest has been given to breast cancer. Studies focused on secondary prevention demonstrate that soya, especially isoflavones, reduce the risk of breast cancer, especially among post-menopausal women. This is the beans or soya which is present inside and dried one. The soya is much useful in reducing the risk of breast cancer. Garlic, we are using garlic in the food and we know that it's highly medicinal active. Garlic, which belongs to Allium genus of vegetables, and its sulfur containing active compounds are protective against the onset and on progression of several cancers. Diallyl trisulfide prevented the development of prostate cancer and the spread of cancer to lungs. They have seen in the transgenic mouse model of prostate cancer. This is the plant, the aerial part, and this is garlic, and this is the structure of diallyl trisulfide, which is responsible for, which is the phytochemical responsible for the action towards several cancer, protective against the progression of several cancers. Green tea, which we have discussed already in detail in various ailments. See here, green tea, polyphenols, catechin, we are talking about catechins also, have been shown to prevent advancement of tumor cells in transgenic adenocarcinoma of mouse prostate model. Green tea, we can say that is very common, which we know. Green tea has been shown to protect the bladder against cell death in bladder cancer, and the antioxidant potency of green tea extract was examined as green tea reduced the oxidative stress induced by H2O2. It's an oxidative agent, or you can say it is a radical. It's normal, abnormal, or malignant human bladder cells. It may cause damage to the cells and proliferation of development of cancer can caused by H2O2. This can be prevented by means of the polyphenols present in green tea have the antioxidant effect by donating the electrons, which we have discussed. It can provide the antioxidant effect and prevent from the cancer. Grape seeds, which we always split it out or remove it, but it has a very good effect of anti-cancer. Grape seed has been demonstrated as being effective to prevent the UV light induced cancer, skin cancer, UV light induced cancer. Grape seeds and grape seeds extract. And in case of control study, grape seed extract has been associated with decreased risk of cutaneous squamous cell carcinoma. Squamous cell carcinoma, as research suggests that grape seed extract greatly reduce the risk of cutaneous squamous cell carcinoma when the extract was compared with a multivitamin. So it has more vitaminic effect than a multivitamin. In squamous cells carcinoma, you can see there is 3 types have been shown in the picture, like squamous cell carcinoma, basal cell carcinoma, melanoma is also shown, the 3 types of skin cancer which can be appeared in this type of forms. It's not risk to the life-threatening for the death, but any of the skin have been getting damaged by this carcinoma, wart-like. So which we can say a polyphenols, grape seeds, what is its present? Proanthocyanidins. Proanthocyanidins have been acknowledged, have been notified, have been proved in the inhibition of lung cancer also. You can see this cutaneous squamous cell carcinoma can come in the forehead, which is an example which I have shown here, skin carcinoma. Ginseng, we are going to talk about the ginseng as a nutraceutical. Ginseng over the years have been used as medicinal plant. Ginseng belongs to the genus Panax and inclusive of Chinese ginseng, American ginseng and NATO ginseng. It has received extensive attention in cancer research involving various cancer types as cancer of the blood, cancer of liver, cancer of colon, cancer of breast, cancer of lungs. Ginseng extract has been suggested to be chemopreventive in the development of colon or colon cancer in vitro and in a mouse model. This is ginseng. This is also another type of ginseng dried which is prepared, that is natto ginseng root. Extract demonstrated an inhibitory effect on colorectal cancers, thereby being preventive in the proliferation of cancer cells. American ginseng extract had a suppressive effect on ulcerative colitis and inflammation of the large intestine and a risk factor for colon cancer when administered to mice and in vitro. It is thus anti-inflammatory of colitis and chemopreventive to the cancer cells. This is Araucaria papacha, Trichopus xylanicus, found endemic to Agastya Hills of Kerala, is used by the local Kani tribes, very famous tribes who talks Malaya language as a health food. They have a health food for getting instant stamina, evergreen health and vitality. The tonic effect of this plant is comparable to that of the famous food drug ginseng. Ginseng in Tamil is Gunasingi, which is available with this traditional local Kani tribes, their healthy food, which is in Agastya hills of Kerala and Tamil Nadu borders. Next one is Rhodella rosae. Rhodiola rosae. Only a few research have involved in Rhodiola rosae in cancer studies. So information regarding its involvement in the prevention of cancer is really limited. Nonetheless, in an in vitro study, Rhodiola rosae extract prevent the growth and the progression of cancer cells in urinary bladder. This is that flower, the lower bunch of flowers, and looks small. The leaves are little ovate and flat, not like, and it looks like growing top with the flower which is shown. It is an Rhodiola rosae Rhodiola rosae is a perennial flowering plant in the family Carassulaceae. It grows naturally in wild Arctic region of Europe, Asia and North America, and can be propagated as a ground cover. Tomato and red pepper. We have discussed about the lycopene. Tomato contains lycopene, which has been acknowledged as being potent and other antioxidants. Studies suggested lycopene is chemopreventive against cancers, including prostate cancer, gastric cancer, breast cancer, and lung cancer. The tomato, which contains lycopene, has been shown to be an effective against liver cancer induced by some environmental carcinogen in a rat model. We should know that diethyl nitrosamine, which is present in initiated and non-alcoholic steatohepatitis promoted early liver cancer, was inhibited in rats by lycopene containing tomato extracts. So they used this synthetic chemical to induce the cancer in the rat, that is diethyl nitrosamine, and this lycopene have inhibited the cell division or the development of cancer. This is, you can see, red bell pepper contain beta cryotoxethin or oxanthin, cryptooxanthin. Beta cryptooxanthin, a compound that your body turns into vitamin A, which is present in the red bell pepper. The crypto or cryptoxethin, beta cryptoxethin, can be converted into vitamin A. Some research suggests that food of beta cryptoxethin may help lower the risk of bladder, lungs, and colon cancer. The last one is dietary fibers and cruciferous vegetables. Dietary fibers is known to prevent that is constipation by increasing the bulk of stool, drawing water into the stool. It has been shown to have significant effects on the risk of colorectal cancer. In the coherent study, dietary fibers was effective in the prevention of colorectal cancer among individuals who were genotypically at risk of the chronic disease. In the colon, and the dietary fibers have direct contact with the walls of the intestine, which in itself reduces the pressure exerted on the walls of the canal. Also, dietary fibers rich diet intake has been found to be associated with the reduced risk of breast cancer in a cohort study with post-menopausal women. This includes broccoli, cabbage, cauliflower, Brussels sprouts, and the greens including kale, turnip, collard, and mustard. In the study conducted in vitro, phenethyl isothiocyanate 3 micro ml or microgram from calciferous vegetables was postulated to be a chemopreventive agent, as it had an inhibitory effect on human breast cancer cell lines. This is broccoli, Brussels sprouts, turnip, kale, collard. These all are some of the examples which we have made. This slide which is again, I am just making you to remember what all the functional foods and the nutraceuticals compounds in the cancer which we have seen is chestnut, berries, soya, garlic, green tea, grape seeds, ginseng, rhodiola rosea, tomato and red pepper, and dietary fibers and cruciferous vegetables. And you know very well, we know about curcumin in curcuma longa, vincristin, vinblastin in vinca, rosaceae, and there are many plant sources which can be used for the treatment of cancer. That is specifically different from the nutraceuticals which are used as a food. We cannot take, but you can turmeric or curcuma longa, which can be used in the food also, so it can be also added in these nutraceuticals. Curcumin everybody knows, which has more anti-cancer effect and various medicinal uses starting from antibacterial effect. And being for this class, thank you very much.